So first, thank you very much, Carol, Jill, for the invitation and the Society. For someone who works on germ cells and epigenetics, it's certainly a wonderful meeting to come to. I'm not a regular attendee, and it's very special to be here for the anniversary. So I've really enjoyed it. So my task is to talk about germ cells, epigenetics, and some possibilities for heritability from some of the studies that we have been doing. So my laboratory's interest has been interested in epigenetics for a number of years, and in particular in regards to preventing birth defects in children. And most of the work that we've done has been on the effects of various genes, diet exposures on the epigenome, and the impact of that on the embryos and the offspring later in life. And most of the work we have done has been on the developing eggs, sperm, and the early embryos. And this is because very dramatic programming, the most dramatic programming during life of the epigenome, takes place as the eggs and sperm are developing, starting in utero, and also during the first 6 days of life. And so I'll show you a little bit of that programming. So it's a time that is particularly susceptible, we think, to environmental exposures. And the one that I'm going to talk about today is maybe unlikely, but it's folic acid. Okay, so you heard a very nice background and history going back a long way from Chris this morning on the epigenome. Here I've depicted in one diagram the different marks that can lead to heritable expression, DNA methylation, histone marks, RNA-based mechanisms. And the important aspect of this is the heritability of these marks, so that if there is a perturbation of a normal epigenomic mark, it can be passed through a number of cell divisions and be inherited. And so most of our studies have been on DNA methylation, and the talk I'll give today will be only on DNA methylation, but you must remember that all of these marks work together, and in fact an environmental exposure could, for instance, affect histone modifications that could then lead to an alteration in DNA methylation, and a lot of the enzymes work together or against one another. So even looking at something like DNA methylation, we have a lot of challenges. And so here I show DNA methylation that occurs on the 5' position of cytosine residues. About 80% of the CpG dinucleotides in our DNA are methylated, and it's about 20 to 30 million, depending on whether you're a mouse or a human. So that's a lot of sites to be thinking about when we're doing studies to see how an environment has impacted the DNA methylome. A lot of the methylation is in retrotransposons that have come into our genomes over an evolutionary time, but also there's methylation within satellite sequences, single copy genes. Imprinted genes are particularly important because there is parental allele-specific methylation, and that if you lose the methylation from one of your parents, you can't make it up after fertilization. So imprinted genes are very important for development, and if the imprints go wrong, there are dramatic consequences. One of the important aspects of DNA methylation and the other epigenomic marks is that they are reprogrammed almost completely at 2 different times of development, in primordial germ cells and in the early embryo in that first 6 days of life. So theoretically, this prevents the accumulation of epimutations across generations, but we're now learning that some can escape. I'll get this figured out. So Today I'm going to concentrate on folic acid. So why folic acid? Folic acid was introduced into the food supply in the 1990s in North America and in other countries to prevent neural tube defects. And it was very effective in doing that. The neural tube defect rates went down. And so if you look now at how much folic acid you get from your diet, it's about 200 micrograms per day. And then during pregnancy, pregnant women will be given extra folic acid at about 400 micrograms a day, and the upper limit is considered to be 1 milligram per day. However, with time, much higher doses have started to be used in a number of settings, including for men with infertility. They've been using doses of 5 milligrams a day, so that's 10 times the dose recommended for pregnancy. And higher doses are also used in when you suspect that there is a high risk of neural tube defects in the pregnancy, but also for other high-risk pregnancies. So for instance, for many women who use assisted reproduction, they will be prescribed these high doses of folic acid. So the questions have come up. Could the higher doses of folic acid be good or not so good? And people have started to look at that. Okay, so why folic acid? So folate provides us with methyl groups for S-adenosylmethionine, which is a universal methyl donor and is important for the methylation of cytosines in DNA. This shows you the metabolic pathway for folate coming in through the diet into the folate metabolic pathway. You'll see here an enzyme, methylene tetrahydrofolate reductase, which I'll talk about a little bit more in relation to the sperm studies. And the methyl groups then get shuttled over to the other side of the pathway, shown here, where they then provide methyl groups for many different reactions, including DNA methylation. So the questions that I want to address today are what is the impact of the high-dose folic acid supplements that are being used on human sperm, and how can we use mouse models to try and help us understand what we're seeing in the human studies, and can some of the exposures we're seeing and some of the effects that we're seeing be transmitted to the next generation? So I'm going to show you very quickly some of the human studies that we've done, but then in more detail the mouse studies to get at the mechanisms. So most of the talk will be on spermatogenesis, but at the end I'll talk about some effects on oocytes. Okay, so just to remind you of the really critical phases of spermatogenesis, we have the primordial germ cells, which divide in utero in the developing testis before birth. And in fact, this is a pretty important time for the acquisition of methylation patterns after they're erased in the primordial germ cells. Following birth, puberty, before that, you have the division of the spermatogonial stem cells, and they maintain the patterns that were set down before birth, and they also lay down some new patterns. And methylation then is remodeled slightly as it goes through spermatogenesis. But there are also some other very important aspects of spermatogenesis, including the very big changes in chromatin that take place, and we know now of histone methylation that is very important in sperm, and that gets inherited in the embryo. Okay, so the two key periods of DNA methylation acquisition are shown here. So initially you'll have erasure in the primordial germ cells, so that's across almost all of the 20 to 30 million sites. And then you acquire methylation in the prenatal gonad in males. and it's maintained postnatally in the spermatogonia as they start into meiosis. Okay, so another way of looking at this and then adding in the female germ cell picture. Here we have development going all the way from before birth, the primordial germ cells, the erasure of methylation, both in the male and in the female, whether you have, depending on whether you have a testis or an ovary. You have reacquisition in the male before birth, but this reacquisition in the female only occurs after birth as the oocytes grow and before ovulation. At the time of fertilization, then, you have both oocyte and sperm-specific patterns, so that for something like imprinted genes, you'll have maternal imprints and paternal imprints. And then at this time, in the first 6 days of life, you have a second reprogramming period where almost all of the methylation again is erased, except for imprinted genes, which must maintain their gamete-derived patterns. And then there is reestablishment of methylation in the embryo and in the placenta to different degrees around the time of implantation. So there are, however, the question that we asked first from our human studies was that if you're giving these men high doses of folic acid, could that folic acid affect the sperm epigenome? So the men that we were looking at were idiopathic infertile men. They're the ones who are often given these high doses of folic acid. And what we did was to give them folic acid daily for a period of 6 months. So this was 2 rounds of spermatogenesis. We collected sperm before they started their treatment and at the end of the treatment. The same time, we did all of the other assays, looking at hormones, B12. We looked at folate to make sure that they had been taking their folic acid. And the most important part of what I'm going to show you here is that we compared the sperm epigenomes. So we looked at DNA methylation using reduced representation bisulfite sequencing. So this is a next generation sequencing based assay that allows you to look at about 3 million, methylation of 3 million CpGs in the epigenome. And on top of this, we added a look at the MTHFR genotype. So methylene tetrahydrofolate reductase is very important for the use of folate in the folate pathway, and there's a very important polymorphism that affects the level of activity of this enzyme, and it can affect disease also. Okay, so this is the enzyme MTHFR. There's a very common polymorphism of this enzyme that changes a conserved alanine to valine. And the men with a TT genotype have 50% reduced activity of the enzyme. And so this is found at about 10% in the population. So it's quite frequent, higher in other populations. And it appears to be overrepresented in male infertility. So in this study that we did, we had 30 men. And of those 30 men, they were about equally divided between those that had the CC genotype, the CT heterozygote genotype, and the TT genotype associated with the decrease in enzyme activity. So I'm just going to show you a summary of the DNA methylation data, and that's shown here. And it's divided up between the first of all, here, all of the individuals, and then the 3 different genotypes. And what you see on the y-axis is the number of differentially methylated tiles. These were at least 2 CpGs had to be affected for a tile to be called. And we had to have at least 10%. So these are significant 10 to 20% changes in methylation. And what you see here is that there are thousands of tiles that are affected as a result of the folic acid, looking comparing the before and the after treatment. So what was somewhat surprising, however, we had expected that folic acid might increase methylation in DNA. In fact, we found both increases and decreases. The increases are the black and the decreases are the gray. But surprisingly, we found more hypomethylation than hypermethylation. And if we looked at our genotype, the TT genotype, this was exacerbated, so that the sperm epigenome was hypomethylated. There was less methylation, and this was unexpected. So then we could look at the various genic regions that were affected, and the types of pathways that came up are shown here. So you'll see neurobehavioral, but a lot of the sites were associated with cancers. So this concerned us that there may be an effect on the sperm epigenome that could be then passed on to the next generation. So how do you test that? It's probably very difficult to do unless you get much larger populations and do the appropriate human studies. And I've been trying to convince my clinical colleagues and epidemiological colleagues that we should be looking at sperm much more often in some of the large studies that are being done. But what we can do is we can go back to a mouse study to try and figure out whether what is the basis of the findings we have here, and is there biological plausibility and similarities of effects. Okay, so what we did was to go back to an inbred mouse model, and with our colleague Rima Rosen, we have a model that represents the MTHFR TT genotype men, and we could use folic acid of 10-fold levels similar to what was used in the human study, and then even higher levels. And these mice were treated for quite a bit longer than in our human study. They're treated for 6 to 12 months. And what we were asking was, if we then go and look at the sperm of these animals, do we see a similar effect as we had seen in the human study? And again, we're using reduced representation bisulfite sequencing so that we can get a genome-wide look across the whole DNA methylome. And the results are shown here, even more dramatic than what we had seen in the human studies. So this is just showing you the number of tiles that were affected, but the light color showing you that the most effect was on hypomethylation. So they had very hypomethylated genomes as a result of the folic acid. In this case, it's the MTHFR ++ genotype with 20-fold dose, and this is the plus-minus equivalent to the TT genotype men with the 10-fold diet. Okay, so why might high doses of folic acid result in more hypomethylation of DNA? So we go back to this pathway. What we know is if you use very high doses of folic acid, you can overwhelm the intestinal breakdown system. You build up the amount of dihydrofolate, and this is a potent inhibitor of MTHFR. So that like having too little folic acid, you don't get enough methyl groups over here. The same thing can happen with too high doses of folic acid. You inhibit the MTHFR enzyme, and this does not allow the methyl groups to get to the other side of the pathway. So how can we prove that this is happening in our system? We couldn't in the men, because most men will not let you go in and do a testicular biopsy. It certainly wouldn't get through the ethics committee, and it would not be clinically allowed. However, you can do this in the mouse. And so what we can ask in the mouse is, do those doses of folic acid affect MTHFR in the testis? And so this is what you can see here, is that there is a downregulation of MTHFR, potentially explaining why we have hypomethylation of DNA. And when we went back to compare the human and the mouse data, we found that there were a lot of common pathways that emerged from the mouse and human DNA methylation data. And interestingly, some of those sites that are known to escape methylation in the prenatal window and in early development were ones that were affected in these studies, suggesting that they might be passed on to the next generation. Okay, so there is a lot of interest in knowing whether or not epigenetic patterns can be altered and the effects transmitted to the next generation. Again, it's easier to do this in an animal study. So what we did was to use these same types of doses we'd used in the postnatal study here and go back and look in a mouse model over a number of generations at both low and at high dose of folic acid and ask whether or not there were adverse effects across generations. So this is the model. These are studies that were published a couple of years ago. And basically what we did was to do lifetime exposure of males. So these are men that were started on their diets. So these were mice that were started on their diets before birth as fetuses. At the time when you erase and then reset methylation, and they were maintained on the treatment until they grew up, at which point they were mated and they produced the F2 and then the F3 generation, but their diets were not continued. So the diet was all the way from all of sperm development from the prenatal to the postnatal windows. And what we did was to look at folic acid control diet. We used 7-fold deficiency. Again, we wanted to see what would happen with too little folic acid. Previous studies from Sarah Kimmon's lab had suggested that there were growth defects and birth defects in the next generation as a result of that dose of folic acid deficiency. We used the 10-fold supplemented and the 20-fold supplemented. So here you can see the F1 litter sizes. So these are the offspring of the exposed males, or the exposed germ cells. And for the most part, the litter sizes were normal. The F1 body weights were normal. However, the sperm counts of these men were lower in both the deficient and in the highest dose of folic acid group. And so then the next question was, it looked like something had happened to the germ cells during that exposure time. Did that result in abnormalities in the next generation if these males who were not on the diets were mated? And so what we see in the F2 is that there was an effect on litter size at weaning in the highest dose group, and there was evidence of postnatal pre-weaning mortality in, again, the low dose and in the highest dose group here. So evidence that those germ cells had affected the next generation. So then we wanted to know, okay, you're treating with these different folic acid diets. Does it affect the sperm epigenome in order to result in these types of effects that you're seeing here? And so what you see here is one of the imprinted genes that we looked at. We looked at a number of different CpGs across the imprinted genes in each of the diet groups. There's very tight results for the control, but there is a spread of the results in almost all of the treatment groups, and you can see the variance here. So that suggested to us that there was an effect on the sperm of the males. And we went on to do reduced representation by sulfide sequencing and found defects in all of our groups, shown here. And what I'm showing is the F1 sperm and the F2 sperm. The F1 sperm are in the white, the second generation in black. And what you can see is that there's differential methylation across both generations. And it appears that there's exacerbation with the highest dose into the F2 sperm. Okay, so basically what we think is happening is that too little folic acid is leading to decreases in methyl groups, and too much folic acid is leading to decreases in MTHFR activity, at the same time decreasing the methyl groups. So it looks like I am getting towards the end of my time. And so this is just a summary of the male results. What we think is happening is that treatments that we have given are through the lifetime of the male germline, all the way from prenatal development through to sperm. At that time, you have not only establishment of erasure and establishment of DNA methylation, but there are also histone marks and small non-coding RNAs. The diets may be affecting both the histone methylation and DNA methylation. But what we see in the sperm is decreases in sperm counts and altered epigenome. What we think is that we may have germ cell heterogeneity and potentially epigenetic instability, and this results in defects in the F2 offspring. So we've also done an experiment on the female germline, and I'm not going to have time to show you that, but I will summarize it. Okay, so what we wanted to know was in the male germline, our exposure had been all the way through spermatogenesis. And so our female exposure, we did just in this period here, prenatally, and not in the postnatal acquisition period. And our hypothesis was that there would not be an effect. And what we got was an effect. So what we think is that in the female germline, these doses of folic acid may be altering histone modifications, which then lead to methylation abnormalities in the female germ cells. So I'm going to go through that quickly. And just summarize, we've used clinically relevant high-dose folic acid and have found altered DNA methylation in human sperm. We think this is relevant because these high doses are used in human infertility treatment, and we're in fact more concerned about its use in high-risk pregnancies because the prenatal germ cells would be affected during that time. Clearly, there was evidence of gene-environment interactions, and this is something that we need to think about for human studies, and we've shown that we can use animal models to try and understand the mechanisms. And so, again, the dose is the poison, and I think this is an example of too much of a good thing, and certainly the NIH has paid attention and had a workshop a month ago to try and look at this problem more clearly. And I'd just like to thank all of the people in my lab. Lundy Lee and Donovan Chan, shown here, were the key people doing these studies, and Mahmoud Rabi. And I have lots of support from our genomics colleagues, our colleagues at McGill and at Health Canada, Amanda McFarland helping us with the folic acid studies. Thank you.